Hello and welcome to Nikolai's genetics lessons, and today's problem is the pedigree below represent family with cystic fibrosis, autosomal recessive genetic disorder. What is the risk that individual 22 will have an affected child if the prevalence of cystic fibrosis is 1 in 2,500 individuals, meaning 1 out of 2,500 individuals is affected. So here's 4 answers to choose from. As usual, I recommend you to pause video here, try to solve this problem on your own first, and when you would be ready, you can run video again and you can compare your answer with my answer and explanation. First of all, as you see with Roman numbers, we designate generations, so generation 1, 2, 3, and if we are looking for individual 2, now we count individuals from left to right using Arabic numeration system. So this would be individual 1, 2, 3, and 4. And we have to find the probability that individual 2, if he would marry and would have a child, that this individual would have an affected child. So, according to this pedigree, as you see, genotype of the father would be small a. If we would say that this genetic disorder is caused by defective allele, this is recessive genetic disorder, so genotype of this person have to be small a. So let's use lowercase a to designate defective alleles. So this person has 2 defective alleles. That's why phenotype of this person is affected. And as for this person here, his wife, her phenotype is normal. So we can say that her genotype would be capital A and capital A. You may also say that there is a slight probability that she can be heterozygous and can be, for example, capital A and small a, and actually this probability doesn't affect our calculations. For example, if this female would be homozygous dominant, so would be capital A, and we know that her husband is small a, as you see. As for the progeny, we are going to get capital A, small a here, capital A, small a here, capital A, small a here, and capital A, small a here. So we can say that person number 2 is obligate heterozygous. So this person have to be capital A and small a. And if we consider that this female is heterozygous, just let's take a look. So if female would be capital A and small a, and her husband is small a, once again we build a Punnett square, and as you see, capital A, small a here, capital A, small a here, small a here, and small a here. So as you see, results are different. If we assume that this female is obligate homozygous dominant, 100% of all the progeny would be obligate heterozygous also. But if we assume that this female is heterozygous herself, we see that 50% of the progeny would be affected and 50% would be heterozygous. So as you see, no matter whether this female is homozygous dominant or heterozygous, if we know that this person is unaffected, then even in this case he obligate heterozygous. If this female is homozygous dominant, he still obligate heterozygous. So now we can move on. when we know that this person is obligate heterozygous, now we can find a probability if he would marry any female, what the probability would be that the child would be affected, when we know that 1 out of 2,500 people in the whole population is affected with this genetic disorder. So in order to move on, our next step would be to find number of heterozygotes in the population, so people who are carriers, and Hardy-Weinberg formula should help us. So frequency of 3 genotypes and 2 phenotypes equal to p squared plus 2pq plus q squared. In this formula, p squared stands for the homozygous dominant genotype, 2pq stands for the heterozygous genotype, and q squared stands for the homozygous recessive genotype. And all these 3 genotypes, when we add them, would equal to 1 or 100%, because 1 here equals to 100%. And as you see, these 2 genotypes would make one phenotype, and this genotype would make another phenotype. So we have, once again, 1, 2, 3 genotypes, but heterozygous and homozygous dominant in simple Mendelian genetics would make one phenotype. And this is going to be another phenotype, so this is going to be healthy phenotype, and this would be affected. And we know that frequency of this affected genotype equal to, and also this means affected phenotype, is 1 out of 2,500. So 1 out of 2,500 people would be affected with this genetic disorder. So how we are going to solve our problem? If we know that q squared equals to this number, so let's use decimal numbers. So q squared equals to basically root out of 0.0004, then we can say what q is equal. Q would equal to 0.02. We just take a square root here, and this is how we are going to get q. And as you see, these 3 genotypes is made by only 2 type of alleles, P and Q. In our calculations, we use A, but we also can change P here for A, but you have to understand that A multiplied by A, so capital A multiplied by capital A, we are going to get A squared. We just used here different notation, so we can change P to A, and P we can change to A capital here, and Q we can change to A small. So if we have only 2 alleles here, P and Q, that makes, or if you like, A capital and A small, so that means that all the alleles A dominant plus all the alleles A small would give us 1. Or, once again, in our formula we are using P and Q. And if we add these alleles that is present in the gene pool, we are going to get 1 or 100%. If we know that Q equals to 0.02, we can easily say that P would equal to 0.98. And when we add these 2 numbers, we are going to get 1. Now when we know P, when we know Q, we can find number of people who are carriers of this genetic disorder in a population. So this part of the formula stands for the number of heterozygotes. So 2pq, 2pq, and this is capital A small a, would equal to 2 multiplied by 0.98 and multiplied by 0.02. So if we multiply these two numbers, we are going to get 0.0196, and now we have to multiply by 2, and we are going to get 0.039, or we can round this number, and this would equal to 0.04. Because all our answers are given in a fraction form, so we can say that this would equal to 1 divided by 25. So let's return. So what we have found the frequency of the people who are carriers. 1 out of 25 people would be carriers. And this is going to be a number of people who would be affected with this genetic disorder. And also, if we add this number and would subtract from 1, this is how we can get a number of those people who are homozygous dominant, or we just can multiply allele A frequency by itself. So two ways. And now we can return to our pedigree and to our problem. And if we know that this person is obligate carrier, and probability that his spouse or mate would be heterozygous. So once again, we know that this person is obligate carrier, so 100%. And if he would met randomly with any female, her probability of being a carrier would equal to 1 over 25. And if we would have two carriers, the probability to have affected child would be 1/4. So I don't have much space left, but one person is a carrier, second person is a carrier, and as you see, according to this Punnett square, capital A here, capital A small a here, capital A small a here, and small a here. If two persons are carriers, the probability to have affected child would be 1/4. So we have to multiply by 1/4. And as you see, probability for this person to have an affected child would be 1 multiplied by 1 over 25 multiplied by 1/4 would be 1 over 100. So probability would be about 1% or 1 over 100, and as you see, this is answer A. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Share this video with your classmates, and see you in the next video. Goodbye.